Hello, I'm Devishi Rastogi, and today we will be talking about proteins, a co-biomolecule. The content includes biomolecules, relevance of proteins, structure and types of proteins, functions of proteins, and study of proteins. What are biomolecules? Biomolecules are life-sustaining organic compounds found in organisms. They support cells, enable metabolism, and regulate vital biological functions. There are four major classes of biomolecules. Carbohydrates. They are the most abundant biomolecules made up of polyhydroxyaldehydes or ketones. They are the energy providers, fueling essential cellular activities. Lipids. They form protective barriers and membranes, insulating organs and storing energy. Proteins, multifaceted in their functions, catalyze biochemical reactions, maintain cellular architecture, and play crucial roles in signaling and transport. Nucleic acids, namely DNA and RNA, they carry genetic instructions and orchestrate gene expression, making hereditary and cellular functions possible. These molecules are not rigid. They exist in constant motion, adapting and interacting within their environment. Biomolecules span a wide range of sizes and complexities. They are primarily composed of carbon, hydrogen, oxygen, phosphorus, and sulfur, often forming covalent bonds. Many of these compounds originate as hydrocarbons, but are chemically transformed by substituting hydrogen atoms with functional groups like alcohols, amines, aldehydes, ketones, or carboxylic acids. Monomeric units such as amino acids, nucleotides, fatty acids, and monosaccharides serve as the building blocks. When polymerized, they form their respective macromolecules, each with specific roles. These molecules are the unseen architects of life, dynamic, responsive, and remarkably complex. Proteins are derived from the Greek word proteos, which means primary or of prime importance. Relevance of proteins. Proteins are indispensable to cellular function due to their immense diversity and adaptability. Their folding and structure stability are highly sensitive to environmental factors such as pH, temperature, and surrounding molecules. This variability allows proteins to fulfill a wide range of roles. Structural proteins. They have evolved to withstand mechanical stress. Materials such as hair, fingernails, wool, silk, and leather are composed of structural proteins adapted to withstand these mechanical stress and protect organisms. Enzymes. Enzymatic proteins, they speed up chemical reactions involved in digestion, blood clotting, replication, transcription, translation. Nearly all cellular transformations depend on enzymes, which enable controlled reaction rates and allow cells to couple energetically favorable reactions with those that are otherwise unfavorable. Without enzymes, cellular chemistry would be virtually impossible. Regulatory proteins. They coordinate the events within and between cells. They turn various cellular processes on and off. For example, turning enzymatic activities on and off in response to environmental signals. Carrier proteins. They help deliver molecules to different parts of cells and organisms, processes such as respiration and metabolism. Carriers, they transport molecules such as oxygen via hemoglobin and lipids via protein-based lipid carriers throughout the bloodstream. Other relevance also includes ion channels, which facilitate ion transport across membranes, signaling which helps cells to perceive and respond to external stimuli, antibodies in immune system detect and neutralize pathogens, even in gene expression, regulating when and how genes are transcribed into RNA and translated into proteins, means they regulate their own production. Altogether, proteins execute a wide spectrum of cellular tasks, from catalysis and structure to communication and defense. Structure of protein. Basic structural components. Amino acids are the fundamental building blocks of proteins, with 20 distinct types participating in the synthesis of proteins in living organisms. Each amino acid shares a core structural framework, an amino group NH2, a carboxylic group COOH, and an alpha carbon atom that connects these two groups. The alpha carbon is a central feature. It is typically chiral in nature. This chirality results in two stereoisomers for each amino acid, but only the L isomer is naturally incorporated into proteins. Each amino acid side chain, that is the R group, determines its chemical traits, allowing classification by size, charge, polarity, and impact on folding. Peptide bonds form the backbone of polypeptides, connecting individual amino acids into long chains that ultimately fold into functional proteins. This covalent bond arises when the carboxylic acid group of one amino acid reacts with the amino group of another, releasing a water molecule in condensation reaction. Specifically, the hydroxyl is removed from the carboxylic group and a hydrogen is removed from the amino group, resulting in the formation of an amide linkage known as the peptide bond. Polypeptide chains exhibit directionality, an essential feature for determining protein function and structure. It has an amino terminus or N terminus, while the opposite end features a free carboxyl group called as the carboxy terminus or C terminus. This orientation matters greatly. The sequence of amino acids must be read and represented from the N terminus to the C terminus. Reversing this direction yields a different polypeptide entirely. Peptide bond is planar and polar in nature, restricting free rotation and favoring the trans configuration due to steric clashes in the cis form. Although the peptide bond itself cannot rotate, the adjacent side chains are flexible, introducing variability in the backbone spatial orientation. These rotational movements occur around two key dihedral angles, phi and psi, collectively known as the Ramachandran angles. These angles dictate the conformation of a polypeptide. Types of proteins. Together, the planar nature of peptide bonds, the variability in rotation, and the chemical diversity of R groups govern the three-dimensional folding into their biologically active forms. There are four structures of proteins. Primary structures. The unique linear sequence of amino acids, each linked by covalent peptide bonds. This sequence is genetically encoded and forms the foundation for all higher levels of organization. The chemical identity and order of amino acids directly influence the folding and biological function of the final protein. Secondary structures. Regular repeating local patterns formed within the polypeptide chain due to hydrogen bonding between backbone atoms. There are two types. Alpha helix, which is the most stable and common conformation, examples including myoglobin and hemoglobin. They are right-handed spiral, giving rise to a tight uniform twist with 3.6 amino acids per turn. Hydrogen bonds stabilize the helix. And the second type is beta sheets, which are almost fully extended chains forming sheet-like arrays, often stabilized by intrachain disulfide bridges. Hydrogen bonds can form between backbone atoms of neighboring strands, which can be parallel, same orientation, or anti-parallel, opposite orientation. Examples, flavodoxin and silk fibroin. Tertiary structures. The three-dimensional shape of the entire polypeptide arises from the folding of secondary structure elements. Structure is stabilized by non-covalent interactions. These non-covalent interactions include hydrophobic interactions, ionic bonds, hydrogen bonds, van der Waals forces, and disulfide bridges in some proteins. This is the most thermodynamically stable conformation. Examples include antibodies. Quaternary structures. Some proteins are composed of multiple polypeptide chains called subunits, which assemble to form a functional complex. They are stabilized by the same non-covalent forces as tertiary structure. Based on the number of chains, proteins can be classified as monomer, one chain, dimer, two chains, tetramer, four chains, and so on. They can also be classified as homodimers, composed of identical chains, heterodimers, composed of different chains. The dissociation of subunits often leads to a loss of protein function. Example includes hemoglobin, which is made up of two alpha helix and two beta subunits. Functions of proteins. Proteins are critical to every physiological process in the body. A deficiency or excess can lead to serious health complications. Starting off with the four main functions, structural functions. Actin and tubulin organize into filaments and microtubules, forming the cytoskeleton, which maintains cellular shape and internal organization, and provides mechanical support and intracellular structure. The image right here you can see is an image of the structural protein tubulin, which is stained red in mouse cells. Proteins maintain the integrity and shape of cells, and without them, structural collapse would occur. Enzymatic functions responsible for driving specific chemical reactions at rapid rates. They're essential for biosynthesis, the process of making new complex molecules from the smaller subunits that are provided to or generated by the cell. RNA polymerase initiates DNA transcriptions. Enzymes also support digestion, the synthesis of macromolecules, and metabolic regulation. Enzymatic proteins such as amylases break down carbohydrates into glucose. These enzymes allow nutrient absorption and fuel cellular metabolism. Gene expression. Proteins regulate their production via transcription factors. These proteins initiate or inhibit gene transcription based on cellular needs. They support the regulation and expression of DNA and RNA via translation, process that forms proteins from amino acids. Transport mechanisms. Proteins embedded in cellular membranes form selective channels that regulate the transport of nutrients, nerve signal transmission, and kidney filtration. Examples include aquaporins for water, GLUT transporters for glucose, and sodium channels for Na+. In blood transport, albumins transport fatty acids, etc. Proteins have a lot more important functions. Proteins act as hormones and regulate other proteins. Example, insulin lowers blood glucose by promoting cellular uptake and glycogen storage. Proteins regulate fluid distribution by maintaining osmotic pressure across membranes. Loss of albumin, a plasma protein, which plays a main role in the above, leads to edema, swelling in various parts, and compromised circulation. Antibodies, immunoglobulins, are specialized proteins that recognize and bind to antigens on pathogens. Once bound, antibodies neutralize or tag pathogens for destruction by the immune system. Neurotransmitters like epinephrine, which transmit signals across nerve cells. Proteins like fibrin, bradykinin, and collagen fibers help in wound healing and regeneration. They also help in nutrient storage. Proteins provide stored amino acids for synthesis, repair, and energy during deprivation. Example, milk has casein, which supplies amino acids during growth. They help in muscular contraction and mobility. Actin, myosin, and troponin work together in muscle fibers. Myosin attaches to actin and uses ATP to pull, causing contractions. Troponin responds to calcium signals to initiate contraction cycles. Last but not the least, proteins are also used for energy generation as a backup system. Proteins are pulled from blood and tissues, risking tissue breakdown when there is a dietary shortage in carbohydrates and fats. Study of proteins. This includes extraction of proteins and then purification to isolate the target protein from a mixture. They undergo analysis and characterization to confirm their identity and functionality. Beginning with extraction, which involves the disruption of cells to release proteins, there are of two types. Mechanical, which includes homogenization, which uses shear force, sonication, which uses ultrasonic waves, and pressure cycling, that applies high pressure for gentle lysis of tough cells. Instrument including barocycler. Non-mechanical methods, detergents, which disrupt lipid membranes, example SDS, organic solvents, ethanol or acetone that disrupt membranes and precipitate proteins, freeze thawing, which uses repeated freezing forms ice crystals to lyse cells. Enzymes like lysozyme break down bacterial cell walls. Then is purification. In centrifugation, it is commonly the first step to eliminate large contaminants from the crude extracts. Its principle is based on density. The supernatant that is collected contains the proteins. Chromatography has four types. In affinity chromatography, it binds the target proteins to ligands on a resin, allowing other components to be washed away, and then elution of protein occurs. It is suitable for proteins with known binding partners or tags. Ion exchange chromatography which separates proteins by charge using cationic or anionic reactions and then is eluted by increasing the ionic strengths. It is suitable for proteins with well-defined charge. Size exclusion chromatography, which sorts protein by size through a column filled with porous beads. Smaller proteins enter the pores and larger proteins which elute faster bypass the pores. It is suitable for separating monomers from aggregates. Hydrophobic interaction chromatography. It relies on the hydrophobicity with proteins binding to hydrophobic groups on a resin at high salt concentrations and eluting as salt concentration decreases. It is suitable for proteins with highly hydrophobic domains. Continuing with purification, the first image indicates how size exclusion chromatography works. Dialysis. The protein solution is placed inside a membrane with selective permeability. Small molecules diffuse out to a surrounding solution. SDS page, which is a gel electrophoresis method, stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, which involves denaturing proteins with SDS detergent and then resolving them by size through a polyacrylamide gel using an electric field. This method not only enables effective separation but also serves as an essential analytical tool. After purifying proteins, analyzing their concentration, purity, activity, structure, stability, and interaction is crucial. Concentration determination. Under colorimetric assays, we have the Burette method, which is where the chelation of cupric ions with peptide bonds in alkaline solution yields a purple solution. It's simple and widely used for plasma proteins but has low sensitivity, making it unsuitable for microgram level estimation. Other methods also include Bradford assay and Basson-Kornick assay. Under spectroscopic method, we have the UV-Vis uh, spectrophotometry, which detects protein absorbance at 280 nanometer due to aromatic amino acids like tyrosine and tryptophan. It's accurate, sensitive, and non-destructive for microgram quantities when properly calibrated. We can even use fluorescence assays, which uses fluorescent dyes to bind to proteins for detection. Under chemical quantification, we have Kejal-Dahl's procedure. Protein samples are digested by boiling with concentrated sulfuric acids at 360 degrees Celsius in the presence of copper sulfate and sodium sulfate, converting nitrogen in proteins to ammonia. The released ammonia is quantified to assess the total nitrogen, and protein content is calculated using the factor proteins are assumed to contain roughly 16% nitrogen. The most accurate and precise method, often used to standardize reference proteins, but it is time consuming, may take several days. Hence, it cannot be used for routine clinical assays. Then comes purity analysis, which has Western blotting, which identifies and confirms the presence of a particular protein in a complex mixture using antigen antibody recognition. It combines electrophoretic separation with highly specific antibody-based detection. During detection, a primary antibody specific to the target protein is applied, followed by a labeled secondary antibody that binds to the primary. Labels may include enzymes, fluorescent tags, or biotin, which generates a visible signal, such as a color, light, or fluorescence, indicating the protein's position. It has high specificity and sensitivity. ELISA. ELISA is a sensitive and quantitative method for detecting specific proteins using antibody-antigen interactions. It's widely used in diagnostics and biopharmaceutical productions, particularly for identifying host cell proteins. The most common type used as an antibody-antigen interaction is sandwich ELISA, where the captured antibody is immobilized on surface of a microtiter plate well. The target protein is added in the image A, in the image which is labeled as A. A second antibody after washing, which is labeled, and then labeled with an enzyme or a fluorophore, produces measurable signals. Structure analysis. X-ray crystallography. Gold standard for resolving atomic level protein structures. Proteins are crystallized and then exposed to X-ray beams. Diffraction patterns are analyzed to reconstruct detailed structural models, delivers atomic precision ideal for drug design, but it requires high quality crystals which can be challenging to obtain for certain proteins. Nuclear magnetic resonance spectroscopy (NMR) offers in-depth information on the three-dimensional structure, dynamics, and interactions of proteins in solutions. It's particularly well suited for small to medium-sized proteins, but it requires large amounts of purified protein and isotopic labelings, example carbon 13, nitrogen 15, which can be resource intensive. Circular dichroism analyzes the differential absorption of left and right-handed circularly polarized light. It is a rapid method to estimate the content of secondary structures such as alpha helices and beta sheets. Monitoring protein foldings, structural transitions, and thermal stability. But it provides low resolution structural data and requires optically transparent buffers. For stability testing, we use differential scanning calorimeter, which gives data on thermal stability and folding, a technique that measures the heat exchange associated with protein unfolding. Mass spectrometry. It is a precise measurement of protein abundance, structural insights, and identifies protein folding, interactions, and post-translational modification. It detects as little as 100 picograms. A mass spectrometer determines the mass of a molecule by measuring its mass to charge ratio. Steps include ionization of molecules, mass analysis, detection of ions and their signals, data interpretation by comparing to standards and databases. With this, we come to the end of our presentation, where the takeaway is why proteins are of the prime importance in every living organism. Thank you.